Hello everyone, welcome back to the class. In previous class we learnt about vesicle fusion. Vesicle is a transport vesicle which has a particular protein inside it, and that protein has to be targeted to a particular destination. So that transport vesicle is budded off from the trans-Golgi network and has different fates based on the sorting sequence present in that protein. So the fusion of transport vesicle with its target involves two types of events. First, the transport vesicle must specifically recognize the correct target membrane. The fusion of the vesicle and target membrane has to happen. So the hypothesis which explains the vesicle fusion is snare hypothesis. What is the snare? Snare is a snap receptor in full. So it is a specific pairs of proteins which mediate the interaction which mediate the vesicle fusion with the target. So this snare receptors are of two kinds based on its location, that is vesicle. If it is in vesicle membrane, if it is in vesicle, so it is V snare. If it is present in the target membrane, it is known as T snares. Based on the identity of a highly conserved residue in it, so it is classified into R snare, that is arginine containing snare, and Q snare, that is glutamine containing snares. Especially in neurons, V snares present on the synaptic vesicle is known to be synaptobrevin, and it is also called as VAMP, that is vesicle associated membrane proteins, and T snares that are designated as syntaxin and SNAP25, that is 25 kilodalton synaptosome associated protein. So next we learned how the process of fusion of that vesicle to the target membrane happens. We learned yesterday. First one is recognition step controlled by RAP protein. RAP proteins are in two forms, that is, one is GDP bound state and GTP bound state. In GDP bound state they are inactive and remain in cytosol. In GTP bound state they are active and tightly associated. What makes this RAP protein to GTP bound state is the particular factor, that is guanine nucleotide exchanger factor. Membrane bound RAP GEF activates the RAP protein. Once in GTP bound and membrane bound state, RAP proteins bind to other proteins called RAB effector. So this RAB effector connects both RAP GTP of vesicle and RAP GTP of the target membrane. The binding of V snare and T snare causes the vesicle to remain bound, and it helps in maintaining the close interaction between a vesicle and the target membrane. After tethering, snare initiates the docking and fusion process. Interacting snares need to separate apart before they can function again. So when the fusion has happened, so this trans snare and V snare will somewhat detach from each other. Most snare proteins in cells participate in multiple rounds of vesicular transport. The complex have to disassemble before the snares can mediate new rounds of transport. So after that, after fusion has happened, then what next? There is NSF. So this has to be NSF. Sorry for the mistake. That is NEM sensitive factor will binds to the snare complex. In this picture you can see here the NSF will bind to what? NSF will bind to the snare complex via adapter proteins. SNAP, that is soluble NSF attachment protein. It is a NSF soluble attachment protein. So it is a soluble ATPase and hydrolyzes ATP to dissociate the snare apart. So this is what we learnt yesterday. This is another image showing that tethering, fusion. Okay, so today we learn a new organelle, that is lysosome. So what do we know about lysosome? That is particularly it is round, it is present in animal cells, and it is a suicidal bag of the cell. We know this much, right? So they contain hydrolase enzymes, which is responsible for hydrolysis, which is responsible for breaking down of the components. So it is a single membrane bound organelle present in animal cells. It is heterogeneous structure and greatly vary in size and shape. It has acidic internal pH, that is approximately 5, and has hydrolytic enzymes like protease, nuclease, glycosidase, lipase, phospholipase, phosphatase, and sulfates. These enzymes are responsible for controlling intracellular digestion of the macromolecule which is happening inside the lysosome. So there is vacuolar H plus ATPase pump, H plus ATPase in lysosomal membrane to maintain this pH, by which the hydrogen ions, H plus ions, will gets pumped into, and it helps in maintaining the internal acidic pH. There are two types of lysosome, remember. Primary lysosome, which do not contain any material for intracellular digestion. Secondary lysosome contains materials that are undergoing digestion or that already have been digested. So lysosomes are responsible for digestion of both extracellular as well as intracellular material. Lysosomal digestion of materials can be classified into autophagy and heterophagy. Autophagy is degradation of cytoplasmic contents within the lysosome, inside the lysosome. Heterophagy, the process by which substances are taken into the cell from external environment and broken down by the lysosome. So this will be dealt in detail in further slides. As we know, it breaks down material and it degrades completely, right? So what are the different types of process involving in degradation is, first let us know the types of lysosome. That is, one is primary lysosome and another one secondary lysosome. Primary lysosome can be said as it is empty, they don't have any material inside it. In secondary lysosome, it has a material undergoing the degradation or already degraded. So these are the two types of lysosome. So lysosome has the capacity to degrade both extracellular and intracellular material. So when it comes to digestion, when it comes to types of digestion where the lysosome is taking part, that is autophagy and heterophagy. Autophagy means it degrades the cytoplasmic component. In heterophagy, the substances are first engulfed into the cell from the external environment and breaks down. Imagine for heterophagy is there is a scenario or this case where the external foreign body has got injected into the cell. So the cell identifies that external body is very much toxic to the cell, or it causes danger to the cell. So cell first engulfs it, and after engulfing it degrades that foreign body. That is an example for heterophagy. But autophagy is a type of process of digestion where the unnecessary material, already used material, which is of no use for the cell, that material are engulfed or are degraded directly with the help of lysosome inside the cell. So first let us learn what is heterophagy. What is heterophagy? Just now I said heterophagy is a degradation happening. First, the substance gets inside the cell from the external environment and then it breaks down. So there is two two term to mention here. First one is phagocytosis. Phagocytosis, and second one is pinocytosis. Pinocytosis is particularly for fluid substance. Phagocytosis is particularly solid substance. You just remember like this. So in heterophagy, there are two different pathways that brings extracellular material to the lysosome. So here from external environment into the cell is happening. The engulfment, the type of engulfment are of two pathways, that is phagocytosis and pinocytosis. Engulfment of large particle is by phagocytosis. The liquid substance which gets inside the pinocytic vesicle, that is known as pinocytosis. First learn what is phagocytosis is. Phagocytosis. So in this phagocytosis, during this phagocytosis, a single membrane phagosome is generated. What is generated? Single membrane phagosome. So this can be phagosome. Phagosome is first generated and this compartment fuses directly with the lysosome. So this fuses with lysosome. This is L, lysosome. Phagosome, when it fuses with the lysosome, it forms phagolysosome. So remember, in lysosome there are hydrolytic enzymes present. The phagosome, when it fuses with the lysosome, so some of the hydrolytic enzymes help in digestion. All eukaryotic cells continually internalize fluid substance is in small pinocytic or endocytic vesicle by the process of pinocytosis, that is different. So phagocytosis involves the particular thing, that is phagolysosome. Whatever proteins or materials present in this phagosome is degraded with the help of lysosome. Okay. So let us see now. So I have said phagocytic cells such as macrophages and neutrophils in vertebrates engulf large particles by the process of phagocytosis. See here, the bacterium is external foreign body where it is engulfing is happening here, that is phagocytosis by a macrophage. So this is pinocytosis. Model of lysosome maturation, how lysosome matures. So imagine in late endosome there is a protein which has been targeted from Golgi. It has come along from the cytoplasm to endoplasmic reticulum and to Golgi, and it is in late endosome right now. The protein is hydrolase. See what happens now. The vesicle has brought that protein into the late endosome. There is degradation happening in the endolysosome. After degradation, there is a digestion of the contents, forming the lysosome. This is the lysosome maturation. In this process, the endocytosed substance first move from the endocytic vesicle to the endosome. So from the vesicle, From the vesicle, the proteins or the substance first moves out to inside the endosome, and at the end of this pathway, the late endosome is converted into endolysosome and lysosome as the result of both their fusion with pre-existing lysosome and progressive acidification. Now let us learn what is autophagy is. Before going to autophagy, let us recall what we learned now. Heterophagy, that is the external substance first engulfed. How this engulfment happens? By two pathways. The engulfment happens by two pathways, that is what? Phagocytosis and pinocytosis. This is the engulfment. After engulfment, what happens? There is degradation and maturation of lysosome. So autophagy is different. It is an intracellular degradation process of cytoplasmic constituent within the lysosome. Okay. First happens, the sequestration begins with the formation of the phagophore. So phagophore forms the autophagosome. So consider, I'll take a blank sheet. Consider there is a phagophore. This is known as phagophore. I'll write PP. Phagophore. Phagophores form de novo in the cytoplasm from a cup-shaped membrane that expands into a double membrane-bound autophagosome. So this followed by some changes, it becomes double membrane-bound and it forms, I'll write it in a different color. So it forms the double membrane-bound autophagosome. I'll write it autophagosome, or I'll write AP. Autophagosome, surrounding a portion of the cytoplasm. It is present in the cytoplasm. The autophagosome may fuse with an endosome. So this will fuse with the endosome. The product of endosome and autophagosome. So the product of fusion of endosome and autophagosome, this whole unit is called amphisome. So starting from formation of phagophore, that phagophore will further develop into double membrane bound autophagosome surrounding a portion of the cytoplasm. That autophagosome will fuse with the endosome. The product of the endosome and autophagosome fusion is called amphisome. Okay, amphisome. Then this amphisome, remember this is amphisome. I'll write it as amphisome, AMS, or amphisome. Okay, AMF itself, AMS, amphisome. So the complete autophagosome or amphisome fuses with the lysosome. So now comes lysosome. Lysosome has what? Lysosome has hydrolytic enzyme which supplies the acid hydrolases, so that whatever present in the endosome will get degraded. Okay, so the amphisome will fuses with the lysosome. The enzymes in the resulting compartment and autolysosome breaks down the inner membrane of the autophagosome. So the lysosome, when it fuses with the amphisome, so the lysosome breaks the inner membrane of the autophagosome and degrades the material. The resulting macromolecules are released and recycled in the cytosol. What did we learn now? First, there will be formation of what? Phagophore. Then it develops into 4 it develops into double membrane bound amphizome or autophagosome. ATP, I'll write it as ATP, autophagosome, or I'll write it as AP itself. Okay, phagophore develops into double membrane autophagosome. This autophagosome fuses with endosome, endosome. This fusion results in the formation of amphizome, and this amphizome will fuse with the lysosome. The hydrolase enzymes will degrade the inner membrane from the autophagosome and degrade the material. The resulting macromolecules are recycled in the cytosol. See here, the process is complete. Here, first engulfment of the cytosol and organelles forms autophagosome. Then lysosome comes into picture. Autophagosome with the lysosome, there is digestion. So these are the electron micrograph showing internalized gold conjugated protein being transported through the structurally diverse organelles of the endosomal compartment. Some lysosomes participate in exocytosis. This enables cells to eliminate undigested contents. For most cells, this happens only when cells are stressed. So there is a process called, till now we learnt endocytosis. This is exocytosis. Exo means out, right? This enables cells to eliminate undigested content outside the cell. For most cells, this happens only when cells are stressed. Okay. They contain a specialized lysosome that have acquired the necessary machinery for fusion with the plasma membrane. There is example quoted here. Melanocyte, melanin producing cells in the skin, has specialized lysosomes which produces and stores melanin pigment. These pigments containing melanosomes release their pigment into extracellular space of the epidermis of the exocytosis. In this image, you can easily differentiate endocytic pathway and exocytic pathway. See the arrows from in the endocytic pathway, it is coming towards the cytosol. Here in exocytosis, it is going out of the cell. There is recycling or transcytotic vesicle. There is clathrin coated vesicle transporting the material into the early endosome for the degradation. Then it moves to the late endosome. After that, late endosome will fuse with the lysosome, forming endolysosome, where there is degradation. Okay, and here in exocytotic pathway, so the Golgi is giving a transport vesicle with the help of regulated secretory vesicles. That vesicle will release that material which has been synthesized by the Golgi to the outer surface or the external part of the cell. Okay, so this was about lysosome, its function, different types of engulfment, types of digestion, that is autophagy and heterophagy, and significance of exocytosis in the cell. So in next class we learn about vacuoles. Thank you.